Next, staphylococcal scalded skin syndrome. Staphylococcal scalded skin syndrome. As the name says, scalds means burn-like disease, which is going to peel off. So you can see this example and easily remember skin is peeling off in this example. This is the scalded skin syndrome that you should be knowing. Okay. Coming to the identification properties of this guy, wherever you see it, it's going to produce that yellow color colonies. Okay, yellow, yellow, yellow. On nutrient agar, yellow color. On blood agar, yellow color, that is beta hemolysis. And on selective media called as mannitol salt agar, again the yellow. Okay, so that's the word right here. Next, moving on to these guys, drugs are important. You should know the drugs here. And in fact, you should also know one more thing. MRSA. Why does the MRSA happen? Because of the mutation. Because of the mutation in the MECA gene. And best test to diagnose MRSA is PCR for MECA gene. Okay. Best test to diagnose this is PCR for the MECA gene. Done. Next, streptococci. Streptococci. When you put the organisms on the blood agar, okay, when you put this organism called streptococci on blood agar, three types of hemolysis can be seen, but actually speaking, only two are important to us because they are the actual hemolysis. Third one is no hemolysis. Okay. If they're doing hemolysis, there are two types of hemolysis. One is complete hemolysis called as beta hemolysis. Okay, this would be your beta hemolysis. Next, incomplete hemolysis called as alpha hemolysis. And if there is no hemolysis at all, that's called as gamma hemolysis, which is a misnomer, right? It's a misnomer. Actually, it's not a hemolysis at all. So technically, this is the thing that you should be knowing here. How do you know this is beta, this is alpha? Color. If you see yellow color here, you should know yellow color is the one for the beta hemolysis. If you see green color here, then this is the one for what? Alpha hemolysis. Done? So that's that you need to know here. Now, who is who? I'm going to say it once over here. Sit back and revise this properly. This topic is important. Beta hemolysis, there are two organisms, group A and group B streptococci. Group A and group B streptococci. And this side, alpha hemolysis, you have these two, one what we call by the name of strep pneumoniae and the other is strep viridans. Okay, so strep pneumoniae and then strep viridans. That is the thing right here. Okay, I hope everybody understood this. Next, let's continue the points here. Once you understood these other things, what is this test for? What is this test first of all? CAMP test. If you're looking at this arrow shape, you have to remember what test right now? CAMP test. So CAMP test is the one that is producing this arrow shape. This is used to identify one organism called as group B streptococci. If the arrow shape is seen, you should know it is for what? It is for the group B streptococci. Okay, so that's about it. It's an identification test of group B strep. All right, let's move on. Streptococcus pneumoniae, please know this guy is the most common cause of pneumonia as well as meningitis in all age groups except the neonates. Okay, all age groups except which one? Neonates. You should know this. Then who is important in the neonates? Klebsiella. Klebsiella organism is the most important for neonatal meningitis. There are others like E. coli, etc. All of them come under one family called as Enterobacteriaceae. Enterobacteriaceae. This is the most common cause of neonatal meningitis in India. This is followed by group B streptococci. Okay, this is followed by what? Group B streptococci. So Enterobacteriaceae members are the most common ones, and then this is followed by the group B streptococci. Okay, so this is the thing that you should all remember. So let's move on. This guy tends to show this colonies on blood agar known as carom coin colonies. Okay, this is simply described as what? Carom coin colonies that you need to know. Okay, next is this thing, doctors. This is your Diplococcus arrangement. Because they are in pairs, they are in couples over here like this. It's called as what? Diplococcus arrangement. This is a feature of this. Okay, so they are roundish. They are arranging in this fashion together, purple and round. It is streptococci. Okay, and if they are in pairs, you should know that this is Streptococcus pneumoniae. Okay, so that's about this guy. Streptococcus viridans. Streptococcus viridans. Now, we have already mentioned about this. These are the most common cause of late onset prosthetic valve endocarditis. Okay, late onset prosthetic valve endocarditis. And the point is, there is one more clinical name for endocarditis, subacute. Subacute bacterial endocarditis also. These are the most common cause. Okay. Native valve, it is aureus. Prosthetic valve, you should divide into two. Early is epidermidis, late is, viridans. So that's something that you should all be familiar with. Okay. Next, pyogenes. Please remember एक point यहाँ पे. Group A strep is a name given to Streptococcus pyogenes. Group A strep is a name given to Streptococcus pyogenes. Now look and understand what I'm telling you right now. Okay. The first thing is this. Based on carbohydrate antigens, grouping is done called as Lancefield groups. That was the scientist. So the groups of streptococci were actually discovered by Lancefield dude. Hence the Lancefield groups, based on what he divided carbohydrate, because there are different carbohydrates in different streptococci. That's how he divided them. How many groups are there? A till V. Group A strep, group B strep, group C strep, like that it goes on. But we just have to know only 1 and 2. Next that you have in this list, the group A. The group A is further divided into types. And what is that types known as? Griffith types. Griffith types. So Griffith types are based on the M protein. Okay, based on the M protein, they are divided like that. Okay. Next, see here. Moving on, you should all know this streptolysin O. This is the toxin that damages the heart, causing a disease called as rheumatic fever. Right? The streptolysin O, it is one of the toxins that causes what? Rheumatic fever, as I'm telling you. And you should know one thing right now. This is the story behind this that you should all be knowing. Rheumatic fever, don't you look for something called as ASO titers? So what is ASO titers, doctors? Antibodies against what? Streptolysin O. Antibodies against this, they are called as, anti-SO. So ASO titers are a diagnosis of rheumatic fever. Now, very important thing that you should pay attention. This scarlet fever is also caused by this guy, scarlet fever, which is going to present. Scarlet fever is going to present as what? It's going to present as basically strawberry tongue. Okay, it's going to present as the strawberry tongue. Scarlet fever is going to present as the strawberry tongue. Okay, So that's about this. Next, probably you all remember this basic idea. Strep pyogenes, sore throat or skin infections are the basic things that can be caused without any more problems. That's it. Most people will get these things, nothing more happens in them. They either get sore throat or the skin infections, and they will be like that. Now, what happens after this is sore throat or skin infection. If some people develop type 2 hypersensitivity reaction, they end up getting a disease called as rheumatic fever. And if it's a type 3 hypersensitivity, they get something called as PSGN, post streptococcal glomerulonephritis. Okay. Post streptococcal glomerulonephritis. This is the word right here. Now, next, that PSGN is the one that is presenting as the cola colored urine. Now, somewhere you must have written this transport media. Pike's media, it is for pyogenes, and this is how you had written it. P for P. P for Pikes, P for pyogenes. Like that, I'll tell you one more. There was something called as Sachs media. Sachs media is for the S organism like Shigella and Salmonella. Shigella and Salmonella, these are the guys, that is your Sachs media. And then we'll be learning Vibrio cholera. Vibrio cholera, it has this thing. Okay. Vibrio cholera. The first two letters, if you take V for the Venkatrama Ramakrishnan, C for the Kerry Blair media. Venkatrama Ramakrishnan, and then the Kerry Blair media. All right. So that's about the transport media you need to know for exam. All right, doctors. Next. Streptococcus agalactiae, I've already told you, this guy also causes neonatal meningitis. It's the most common cause of neonatal meningitis worldwide, but in India, they are the second most common cause. I repeat last time, in India, it is Enterobacteriaceae members like Klebsiella most commonly, followed by group B strep. Okay, followed by the group B strep like agalactiae. They are the ones to cause neonatal meningitis. Okay, so that's the word that you should all be thorough with. Next, gram-positive bacilli. This is your next one in the list here, gram-positive bacilli. Since we are switching gears, it's a new thing that we are coming down to. What is gram-positive bacilli? You have to remind yourself, they are rod-shaped like this. Please remember who are the organisms to look like that. A, B, C, D, L, M, N, right? This mnemonic, A, B, C, D, L, M, N. Okay, so now we are going to study these guys right now. Bacillus. Bacillus mein, what you should be remembering here, doctors. Bacillus anthracis, this is one of the organisms that is considered to be obligate in nature, obligate aerobe. Obligate aerobe, that means it has to has oxygen. If it doesn't easily survive. And whenever bacteria find unfavorable conditions for themselves, they will form spores. They will form spores. Okay, is that understood by all of you? And how are the spores of this guy described as? Non-bulging spores. They're described as the non-bulging spore. You can see the picture right away here. Okay, this is the non-bulging spore as such. This is an organism you're seeing like this, one organism, two organism like this. They're looking like a bamboo stick. We know this appearance also, right? Bamboo stick appearance is seen with Bacillus anthracis. Spores, if you notice, they are just confined inside the organism. They are not popping out. Hence, they are called as non-bulging spores. Done? Next, if you see here, Clostridium point of view, Just look at this one. Do you see very clearly this is a bulging spore? Yes or no, right? So rod-shaped organism. This is also a rod-shaped organism. You might be seeing this bulging spore like this. So these bulging spores belong to Clostridium. Clostridium, as such, they have bulging spore. Non-bulging spores are a feature of Bacillus. Take it. Now the disease of this anthrax, you should all start from this picture. This is the eschar, black color necrotic area. That is the eschar. If you see this eschar, you should know they are talking about Hyde Porter's disease. I told you, right? If you're transporting skins of the animal, if you're transporting skins of the animal, then you're very prone to getting this infection. Okay, is that understood by all of you? So this is the one. Next, moving on, pulmonary anthrax. This is the next one in the list here. Pulmonary anthrax, which is also called as Wool-Sorter's disease. This is the one that is fatal because it's causing pneumonia. And how do you get pneumonia? The organism or something should get into your lungs first. That means the route is inhalation. Inhalation of spores is the route to get this pneumonia. So Hyde Porter's is the skin form of anthrax. Wool-Sorter's is the fatal form, that is the pulmonary form of anthrax. Okay, is that understood by all? Next. Please know, almost organisms who may be causing pneumonia or meningitis may have capsules. Capsule is one extra feature that is present in those bacteria that may actually, cause meningitis or pneumonia. Most of the organisms will have that. Okay, is that clear enough to all of you? Now, try and understand this. The capsule is made up of lipopolysaccharide for almost all organisms. So all organisms have lipopolysaccharide or the sugar capsule. The only one to have a protein capsule, it is this guy called as anthrax. Okay, so protein capsule is present only in the anthracis. Now the point is the tests. For the LPS, the test is Quellung's reaction or latex agglutination test. Okay, latex agglutination test that you should know. And then for the protein, you have McFadden's reaction that you're seeing here. Okay, McFadden's reaction. So that's about it. I have already told you the appearance, very, very important here. It is this one, what we call as bamboo stick appearance. Okay. Next, Bacillus cereus, one word, Chinese fast food poisoning. It mimics Staph aureus in every aspect, fast as well as heat stable. So that's all you're going to know here. Okay, it's fast as such. Next, Clostridiums. I already told you about the bulging spores of these guys. See, there are 4 Clostridiums that you should all be knowing: tetanus, botulinum, perfringens, difficile. Okay, I repeat. One is tetanus, botulinum, perfringens, and difficile. First two are quite opposite in presentation. One causes contraction, second causes relaxation. So you know, tetanus causes contraction, no intro needed. How does it produce contraction? That's a very important thing. See, it inhibits the release of GABA or glycine. What is the job of GABA or glycine in the body? To relax the muscles. The job is to relax the muscles. So what I'm saying here is that here's where it is going to contract now. So how does it do that? It blocks GABA or glycine. GABA or glycine should relax the muscle. But if you block GABA or glycine, you will get a contraction. And the name of this toxin, it is known by the name of tetanospasmin. Okay, the name here is what? Tetanospasmin. That's the thing that you should know right now. Tetanospasmin. Okay. Next thing that you should all be knowing over here is this, the clinical features. Clinical features, okay? They are just contractions with fancy names. The main thing being the difficulty in opening the jaw, called as lock jaw, as if the jaw is locked, right? That is known as what? Lock jaw. Medically, it's known as trismus. Okay. Next, we have botulinum. The next in the list, what we have here is botulinum. Okay. So what you're supposed to know is botulinum relaxes the muscles of the What does acetylcholine do? Acetylcholine contracts muscles. Acetylcholine simply contracts muscle. So if you block acetylcholine, it will relax muscles. Let me give you time to process that. What is the job of acetylcholine? Contracts muscle. So if you block it, now the muscle should relax. And because of this relaxation only, unlike unlike all the other food poisoning which present with diarrhea, this one is going to present with constipation. Okay, this one's going to present with what? Constipation. Is that clear enough to all of you? Instead of this diarrhea, this is going to present with constipation. It's a food poisoning. Botulism is primarily a food poisoning that you should all be aware of. Okay. Now, next, moving on. If you're talking perfringens, okay, if you're talking perfringens, that is Clostridium welchi. All right. You all may have learned already, it causes gas gangrene, right? It causes this gas gangrene. And you should know the gas gangrene is caused by the toxin, what we call as lecithinase. Okay, it is caused by this toxin, what we are calling by the name of what? Lecithinase. Okay, now that if you understood this much, now just see here. There was a mnemonic we wrote, superstar NTR, remember? So this is what you have to remember for that. Stormy fermentation in the milk media, that's your S. A test for the toxin. This is the test for the toxin called as Naegler's reaction. Okay, Naegler's reaction. This is the thing. Next, target hemolysis. This is described as the target hemolysis because you're seeing this center that is darker like this. And this target hemolysis comes due to the factor called as theta factor. That target appearance, it comes due to the theta factor in that. And last May, guys, reverse camp test. Okay. Last May, you have this thing known as reverse camp test. So that's about it. This is the thing you should be knowing here. Next, you have Clostridium difficile organism. Two words, very, very important. Pseudomembranous colitis. Pseudomembranous colitis or antibiotic associated diarrhea is caused here. So what's the word? Pseudomembranous colitis or antibiotic associated diarrhea. This is the word you should be knowing. Now, please know this diarrhea, if it happens, usually what we do is culture, etc. is more important. But in this one, the toxin demonstration would be more important. A better test here would be the toxin demonstration. Is that clear enough to all of you? Toxin demonstration would be much more important. Okay? Now, that's about that. You must know the antibiotics here, like that we have learned in the class, right? Those antibiotics are important again. Next is diphtheria. In relation to the diphtheria, you should know this disease is not directly by the organism, but rather due to the toxins, toxemia. Okay, it's rather due to the toxins, that is the toxemia. It works by inhibiting protein synthesis. The toxin of diphtheria works by inhibiting protein synthesis. So you should just remember one word, what I'm telling you, CDEF. Do you remember this mnemonic? Corynebacterium diphtheriae inhibits elongation factor. CDEF is Corynebacterium diphtheriae inhibits elongation factor. Now, you probably remember these things easily. This is your respiratory diphtheria presenting with this membrane, right? Dirty pseudo membrane. Everybody may have recognized this already. This is the diphtheria that you should know about. Okay. Bull's neck appearance, you all know. That is another thing, right? Bull's neck appearance, another thing that you see over here. Now, think about it once you have learned this clinical presentations, how to identify this organism. The identification of organism here is this classical Chinese letter pattern. Okay, this classical Chinese letter pattern, this is something that you should be knowing here. Okay, now what is this stain over here? It is a special stain by the name of Albert stain that has ability to stain even the granules. It has the ability to even stain the granules. What are these granules called as? Metachromatic granules or Babe Ernest granules. Okay, so usually this arrangement, like in a shape of V like that, it is known as cuneiform arrangement. Okay, this is known as the cuneiform arrangement. Okay, so this is all that you need to know with this. Now coming to the diagnosis aspects, we're going to culture this guy. Two options available. One, I can culture it on Loeffler serum slope. This is the one. Or on potassium tellurite agar, PTA, potassium tellurite agar. So one, Loeffler serum slope, another potassium tellurite agar. Know the advantage and disadvantage of both. Loeffler serum slope is serum. You should all know when in culture media, if you add blood, serum, or egg, it is called as enriched media. It is called by the name of what? Enriched media. Blood, serum, or egg. If you add this, it is known as what? Enriched media. Okay, if you understood this. Now, know the thing, the next part, what I'm telling you here. If you know that is the enriched media that you have learned about, see, enriched media is liked by everybody. That means any organism can grow as such. Enriched media is liked by pretty much everyone, pretty much by everyone. Okay? So now, if you got this, it's not only for diphtheria, that's the disadvantage. It's not a selective media for diphtheria. Loeffler serum slope, since it contains serum, it's a enriched media. It allows the growth of unwanted organisms also. Okay, that's the first thing. Then what is the advantage of using this? It's fast. The growth is faster. The visible growth is seen within 6 to 8 hours only. Within 6 to 8 hours, we see the visible colonies of this organism. Okay? Whereas on the PTA, potassium tellurite agar, this guy called as PTA, what you're supposed to know is the black colonies, what you're seeing, that's the identification. It's the best media. Why am I saying it's the best media? It's only for diphtheria. Potassium tellurite agar, it's only for diphtheria, so it's a best selective media. What is the disadvantage? Just like any culture media takes time. How much time does it take? 2 days, doctors. How much time does culture media take for the reports to come back? 2 days. This guy also is going to take 2 days. Okay. So disadvantage is it's slow, but the good thing is it is the selective one only for diphtheria. What is the problem with Loeffler serum slope? You will have unwanted growth also. It's not selective for diphtheria only. Okay, so that's about that. Baki, moving on to the next part here. See this organism Mycobacterium. So Mycobacterium pe, let's jump to this diagnosis aspect itself, doctors, which is the most important point here. See, you should all know one thing. First, we should all collect a sputum sample. Spot and early morning. This is the preferred one. Okay. Spot and the early morning without any food or anything. This is the sample we will have to collect here. And we will have to more or less clean this sample called as homogenization by using Petrov's method. By using Petrov's method, we are going to clean this sample. Think of it like that. Once the sample for TB is available, all you're going to do right now is stain this, as such. One of them is known as acid-fast stain, and the second is known as auramine rhodamine stain. So these are the two things right now. One is the acid-fast stain. Second is the auramine rhodamine stain. Auramine rhodamine stain. Acid-fast stain, right? It is also called by the name of Zeal-Nelson. Okay, it's also called by the name of Zeal-Nelson or Quignon method. Okay, Zeal-Nelson method or then the Quignon's method. This is just another name for this thing. Okay, auramine rhodamine fluorescent dye. This is the fluorescent colors. That's the only difference. The TB on acid fast will appear which color? Pink color. And then it will appear in fluorescent colors as such in the auramine rhodamine. The problem I told you with the culture is the LJ media, the culture takes around 6 to 8 weeks. The culture takes around 6 to 8 weeks for the growth of this organism. All right. Next, if you talk about the best diagnosis that is done for the TB these days, you should all know it is the PCR or the NAT. Okay, PCR or the word NAT. Now, what is the advantage of the PCR or the NAT? Within 2 hours or so, this is the turnover time for this organism. It is just 2 hours. PCR or NAT takes only 2 hours as such for the report to come out, whether you have TB or not. Okay. Next, once this guy has done this, it also does something else. While it is diagnosing TB, it also checks for one of the mutation called as the RPOB gene mutation. RPOB gene mutation. If this mutation is present, okay, if this mutation is present, yes, mutation is seen. It means rifampicin resistance is present. This R for R is for the drug rifampicin. We are checking if rifampicin resistance is present over here. Okay, so if rifampicin resistance is present, more or less, this would be the MDR TB. Okay, MDR TB, multi-drug resistant TB. No, if this is not present, mutation is not present, it's a good news. It is simply known as the drug sensitive TB. This is known as the drug sensitive TB. So one, it is MDR TB. Second, it is drug sensitive TB. Okay, what if it is MDR TB now? If it is MDR TB, now you want to know which drugs will work, which will not work, for which you have to do a test called as LPA, line probe assay, for which the results roughly come in 2 to 3 days max. Okay, so technically speaking, now if you're having a drug sensitive TB, your diagnosis would be done within 2 hours, confirmed. Whereas if you have drug resistant TB, your diagnosis would be done in 2 days, because I'll have to do line probe assay to know what drugs will work and what will not work. Next and the final, you have diagnosis of latent TB, where I told you this is not that high yield of a thing. Mantoux test and IGRA, these tests are solely based on previous exposure. If you're already previously exposed, that is having latent TB, if you're previously exposed having latent TB, whenever you do any of this test, Mantoux or IGRA, your body will give a strong immune reaction against this antigens you're injecting from outside in this test. Let it be Mantoux, let it be IGRA, the immune response is going to be quite strong if your body is having latent TB, because your body has made something called as memory cells against TB now. If you are having latent TB, what has your body made against latent TB or any TB organism? Memory. So from outside, if you're injecting some antigens of TB, like, tuberculin antigen, etc., that means your body will give a very strong reaction. That reaction is something that we are going to measure. Okay. That reaction is something that we are going to measure as such. Okay. So that's about it. That's your Mantoux test or the IGRA. All right. Next mycobacterium, it is your leprae. Hansen's bacilli is the name, hence leprosy is also called as Hansen's disease. Please know, symptoms of leprosy would be confined to two parts, skin and nerves. Okay, skin and nerves. These are the two things to which this symptoms of leprosy would be confined to. Okay. Now try and understand, when the skin is involved, it is going to present as hypopigmented patches, and the nerve will lead to loss of sensation, what we call as anesthetic patch. Okay, it would be called as the anesthetic patch. So that's about it. Next, the most common nerve involved here is ulnar nerve, that you shouldn't forget. Okay, the most common nerve involved here is ulnar nerve. So that's about this guy here. See, there are two types of leprosy. If you are having a strong immunity versus weak immunity, you will develop this leprosies as such. If your immunity is weak, quite reasonable, bacteria would be more, multibacillary leprosy. And if the bacteria are more, the disease is more severe. The disease is more severe. And if you have a severe disease, multiple lesions of the disease would be present in the body. Okay. Everything opposite in tuberculoid. Strong immunity, less bacteria, less is called as posse. Mild disease, since the disease is mild, you will see fewer lesions in the body. Very few lesions would be seen in the body. And lepromine test, if you talk about, lepromine test is positive with tuberculoid leprosy, and it is negative with lepromatous leprosy. Okay. Because again, strong immunity is needed for hypersensitivity reaction like lepromine test. It's a type 4 hypersensitivity reaction. Okay. So that's about this organism as such here, that is leprae. Now, throughout the course of the disease, some people will get some reactions, hypersensitivity reactions called as lepra reaction. Okay, lepra reaction. So these are the two lepra reactions as such here. Dono ka treatment, if this lepra reaction happens, you have to give steroids to suppress the immunity because they are hypersensitivities. Okay, and that was a recent question as such. Please know, steroids are supposed to be given in this treatment. Next, watch this point, smear microscopy. If you collect a sample, right? If you collect a sample, you will always collect a sample where the organism would be expected. Yes or no? So you should know one thing right here. If the samples are supposed to be taken, one should be from the skin I should take, because that's where the organism can be found. Second is from the nerves. Second is from the nerves. And you should all remember, the nerve sample is taken from cutaneous branch of radial nerve. Okay, cutaneous branch meaning sensory nerve. This is where we are taking the sample from. It is basically a sensory nerve that you should be knowing. Okay, why? Because during the nerve biopsy, we may end up damaging the nerves. And I don't want to destroy a motor nerve like ulnar nerve. Because if you lose the function, motor function, it becomes difficult to do your everyday life. If you lose hot or cold sensation, I can function, I don't mind, but I can do these things. I can lift anything I want to. So the point is this, right? Cutaneous branch of radial nerve. This is the main thing that you should know. And as usual, like TB, you can do the acid fast staining for this guy also. Okay, acid fast staining can be done for this organism also. Please know this point here. This one grows in this animal called as armadillos. TB can be cultured on LJ media. This is the LJ media. This is tuberculosis organism. Green colored bottle. This is LJ media. It's an artificial media. But leprae, the brothers of TB, will not grow in artificial media. You have to use this animal. What is this animal known as? Okay, armadillos. So this is the word that you should know as such. The last part of the bacteria is gram-negative cocci. Now, I would want all of you to know this thing. Since we are talking cocci, they're going to be round. And since we're saying gram-negative, they're going to be pink. Okay. So gram-negative cocci. You have two guys in here, Neisseria meningitidis and Neisseria gonorrhoeae. Okay. Meningococcus and gonococcus. These are some other names of these guys. All right. And one of them is kidney shaped, as we have learned before, and the other is described as the lens shaped organism. Okay. Now, since one of this guy, it causes meningitis, it will have a capsule and the other will not have a capsule. So who's causing meningitis? Neisseria meningitidis, hence it is capsulated. And these becomes important identification properties for them, that Neisseria meningitidis ferments, maltose and glucose. Just a hint, M for meningitidis, M for maltose and glucose, whereas G for gonorrhea, G for glucose only. Okay, that's the word right here. Okay, so Neisseria meningitidis or meningococcus, please remember, this guy can actually produce shock. It secretes endotoxin that produces shock. Okay, so that's your Neisseria meningitidis. It produces shock, secretes endotoxin. Now, what are the risk factors here? The risk factors you should remember, it is nothing but MAC deficiency that increases the risk of this organism. Apart from the routine features of meningitis that you see, like nuchal rigidity and all, this is the guy that presents with additional thing, that is the rash. So rash presence here, nuchal rigidity is one thing. Apart from this, there is presence of rash. Now, this rash, if you see, you should diagnose right away. This is Neisseria meningitidis. It's a petechial rash that can happen on the body. Complications. If we talk about complications of this organism, you should know Waterhouse-Friderichsen syndrome. Remember this name? Waterhouse-Friderichsen syndrome is nothing but shock. So any meningitis plus shock, if they mention, answer is Neisseria meningitidis. Okay. Now coming to these things, gram-negative organism, it is diplococci, all that you know. I have told you for capsule, latex agglutination test would be done. Okay. Now, when you do the culture by taking CSF or blood sample, you have to use the agars like blood or chocolate agar, which are enriched media again, not for one organism. Many organism grows on this. Selective media, it is only for Neisseria. Okay, it is only for the Neisseria. So that's about this thing. Okay. Now here we go. One test that you should know. Gram-negative organisms are further classified like this: gram-negative oxidase-positive organisms, gram-negative oxidase-negative organisms. And if you see oxidase-negative organism, you should know they are talking about a family called as Enterobacteriaceae family. Okay, they're talking about Enterobacteriaceae family. That's the word as such that you need to know. Okay. Next thing, if we move from here, Neisseria gonorrhoeae, it is just going to produce discharge. Okay, Neisseria gonorrhoeae is going to produce a discharge. If it's a male, the discharge is due to urethritis, which presents at a purulent discharge from the urethra. Okay, it causes urethritis. Next, please know, even if the urethritis is not caused by gonococcus, it's called as non-gonococcal urethritis. NGU stands for non-gonococcal urethritis. NGU, that's the word. And you should know who's the agent to do this. Chlamydia. Chlamydia is the organism to do the non-gonococcal urethritis. In fact, everything you're studying right now in gonococcus applies to chlamydia also. Chlamydia also does all these things. So I'm not going to teach you this again. So chlamydia in males causes urethritis. In females, Neisseria gonorrhea as well as chlamydia causes cervicitis. They both cause all these things written here. I'm sure you remember this violin string complication we had learned, Fitzhugh-Curtis syndrome. Okay, Fitzhugh-Curtis syndrome. This is the thing that you need to know, which is the complication in the females. And in the babies, ophthalmia neonatorum. Okay, ophthalmia neonatorum. This is the thing happening in the neonates, hence the name neonatorum. It's a conjunctivitis that is manifested as a discharge through the eyes. Okay. Next, if they ask you the selective media, as usual, nothing changes. Thayer Martin or the New York media, as I told you, this is what you need to know for this organism. Next, this is the organism that ferments maltose. Sorry, this is the organism that ferments glucose only. Okay, it ferments glucose only. So that's the keyword as such to be known here. Okay, let's move on to the next part. Gram-negative bacilli. Okay, gram-negative bacilli. What you need to know, Enterobacteriaceae. See, this family of Enterobacteriaceae, they are called members of Enterobacteriaceae because they all have some common properties or common features. And that's the mnemonic you can see right there. Catalase positive, oxidase negative, motile, ferment sugar, etc., etc. So these are the points that you need to keep in mind. Now, just a quick recap. This agar, hopefully you recall the picture. This is known as McConkey's agar. And why do we use McConkey's agar? To differentiate between Enterobacteriaceae members, which are lactose fermenters and lactose non-fermenters. Okay, so simply there are two names here, lactose fermenters and lactose non-fermenters. The lactose fermenters are those that are going to appear pink in color. And if you see pink color colonies on the McConkey's agar, if I'm telling you, you should know the answer to that is either it is E. coli or Klebsiella. Lactose fermenters are who and who, doctors? E. coli and Klebsiella. Lactose non-fermenters, agar aapko yaad ho, this is the mnemonic you have written in the notes, called as SHIPS. Shigella, Yersinia, Proteus, Salmonella. So these are the names: Shigella, Yersinia, Proteus, and then Salmonella. That's the mnemonic called as SHIPS over here. Okay. Now, moving to the next thing. If I show you this picture and ask you pink color colonies on McConkey's, who do you think this is? Either E. coli or Klebsiella. Okay. Next. Diseases caused, do keep in mind, E. coli is also a very important cause of neonatal meningitis. In fact, I have told you before, Enterobacteriaceae members are most common cause for neonatal meningitis in India. Okay, it's the most common cause of UTI. It can cause a lot of variety of conditions that you can see right here. Okay. Now, diarrheal agents may, there are top two that I'm going to cover right now. ETEC and EHEC. These are the two diarrheal agents you should be knowing, ETEC and the EHEC. Now, what is the story? ETEC, T for T. Please remember, it's the most common cause of traveler's diarrhea. T stands for toxogenic. Enterotoxogenic E. coli, ETEC. Most common cause of traveler's diarrhea. How does this diarrhea work is that it increases a second messenger called as cyclic AMP primarily. Okay, assume if the cyclic AMP goes up, you should all know this is going to manifest either as edema or watery diarrhea. Okay, if cyclic AMP is increased by bacterial toxins, it is going to manifest either as edema or the watery diarrhea. Okay, is that understood by all of you? So just remember this point here. Okay. EHEC, on contrary, H for hemorrhagic, meaning bloody diarrhea. This is a watery diarrhea, traveler's diarrhea, whereas this is a bloody diarrhea. And EHEC, important that you should know is the most common serotype is O157H7. Okay. The most common serotype that you should be knowing here is O157H7. Memorize this mechanism of toxin right now. This one inhibits 60S ribosome. It produces the toxin, Shiga-like toxin, which inhibits 60S ribosome, which in turn inhibits protein synthesis. Okay. And it's done by another organism also, Shigella. That's why the name is Shiga-like toxin. Are you seeing the word here? Shiga-like toxin. So you have Shiga toxin and then the Shiga-like toxin in this list here. Okay. Hope everybody understood the point that we are mentioning here. Next thing is this. Infective dose. It is very less. Only a few bacteria can actually initiate the infection. As you can see, only 10 to 100 bacteria, they're able to produce this infection, right? As you're seeing, only 10 to 100 bacteria, they are actually producing the infection here. That's a very important point. In fact, whatever you are studying right now, apply the same for Shigella. Even the complications, EHEC, what are the complications? Hemorrhagic colitis and hemolytic uremic syndrome. Hemorrhagic colitis and hemolytic uremic syndrome. That's about that. Next, Klebsiella, one-liner. Klebsiella pneumoniae causes pneumonia with this sputum, special sputum, red currant jelly sputum. And coming to this organism, Klebsiella granulomatis, this is going to cause granuloma inguinale or venerum. Okay. The more important name of the disease is donovanosis. Right. I have emphasized this before and I'll tell you this once again. Donovanosis has no relation to Leishmania donovani whatsoever. Okay. It has no relation to Leishmania donovani. Leishmania donovani. Okay. Now, how does this disease present? Donovanosis mein you should be easily recognizing a beefy red ulcer. Okay. Donovanosis disease has beefy red ulcer. This is what you should know. And diagnosis is done by these bodies known as Donovan bodies that has this bipolar appearance or safety pin appearance. So that is inside a macrophage, you are seeing this organisms having what kind of appearance? It is having this appearance that is the right beefy red, the bipolar or safety pin appearance. Okay. Next organism, Shigella. Now, what's the deal with the Shigella? Please know it is the same as EHEC. All the properties that I told you here, all the properties I told you here in the EHEC, they apply here also. Okay. Now, get an idea. The only one or two things that I would want you to revise here is this parts. These are the culture media that you need to keep in mind for growing Shigella and Salmonella. Everything is same for Shigella and Salmonella here. Okay. In fact, I told you today Sachs media is for Shigella and Salmonella, right? So this is the thing that you need to have as such. Okay. That's Shigella. Next, Yersinia organism. First, know Yersinia pestis causes this disease known as plague. Yersinia pestis is the causative agent of this disease called as plague. This would be an important topic. Please know that the reservoir for this, it is the rodents or rats. Okay. Rodents are the ones that can spread this disease. And what else should you know is how does this disease from the rats come to the human? It will come by the bite of this insect, what we call as rat flea. Rat flea is going to spread this disease known by the name of Xenopsella chiopis. Okay. Next, disease point of view, you have bubonic plague, buboid, lymph node involvement is there, giving you black color in the respective area that was involved. Pneumonic plague, the one that spreads from human to human from now because it's pulmonary thing, that is pneumonia. And this becomes very, very fatal because it's a direct spread from human to human, very easy transmission. And septicemic plague, this is the one that causes gangrene. Okay. Now, diagnosis, just like the others, what you saw, it shows the same bipolar or safety pin appearance like this. And one characteristic growth called as stalactite growth that I have told you, it is like things that are hanging from the roof. This is the stalactite growth that you're seeing here. Okay, doctors. So hope this is clear enough to all of you. This is the stalactite growth. Okay. Proteus may, two things I will tell you here. Proteus you should know for UTI with alkaline urine. Alkaline urine is not seen with all UTIs. It's with this guy because of urease positivity. Whenever you hear the word urease positive, you should know they are mentioning about this thing, that is alkalinity. Urease enzyme converts urea to ammonia, and ammonia is alkaline in nature. That's why the urine has become alkaline. And because of the alkaline urine, this person will now start to form stones. Alkaline urine precipitates to form stones. So that's the key word right here. Next, diagnosis point of view. Please just watch here. It produces this motility, what we call by the name of swarming motility. And this is the one which is described as swarming motility. Okay. So this ripple-like effect, what you're seeing, it's described as the swarming motility. Done. Remember another heterophile example here, Wheel-Felix reaction. Wheel-Felix reaction. It is actually the antigens of Proteus, the topic we are studying, will resemble the antigens of Rickettsia. And not all Proteus, there are multiple strains, only these strains will resemble. Which ones? OX2, OX19, and OXK. Only these strains, they will resemble, the rickettsial ones. Okay, so that's about that. Salmonella, nothing much. You all know it causes typhoid. Okay, they will not give you stepladder, pyrexia, all these words. Just remember this thing. Typhoid may, if a question is asked, more or less, they will ask you about this. That is the BASU. Okay, diagnosis of typhoid week-wise, first week, second week, third week, fourth week, which test you have to do. That's what you have to know. Mnemonic here is what? BASU. Okay. So in the word BASU, first week, if you suspect typhoid in someone, we will have to perform something called as blood culture. Okay, blood culture needs to be performed. Second is antibody detection test. Antibody detection test, that is the agglutination test here. Okay. Third is stool culture. Fourth is urine culture. So you have seen all these culture words. The most popular test that everybody knows is Vidal test that they can ask you here, right? What is the name of the test here? It is called as the Vidal test. Now, what's the story in the Vidal test that you need to keep in mind? First, are we detecting the antigen or are we detecting the antibody in this? The answer is we are detecting the antibodies. Okay, in this, what are we detecting, doctors? We are detecting the antibodies. Vidal test detects antibody. Antibody against what? Antibody against the Salmonella organisms. There are three Salmonella organisms. First, Salmonella typhi. Second, paratyphi A. Third, paratyphi B. So these are the three as such that you need to know: typhi, paratyphi A, paratyphi B. Now that if you have understood this, please pay attention. Each of them will have antigens, okay, called as O antigens, and O antigens are same in all the three. That means if you even detect antibody against O antigen, you will not be sure if this antibody against the O antigen belongs to typhi, paratyphi A or paratyphi B, because the O antigen is exactly the same in all. For this also, for this also. That means O antibody does not tell you who is causing typhoid, but it tells you you have the disease typhoid. Because why is the O antibody formed? Because you are infected with one of the three organisms right now. Okay, then comes the H. The H antigen of typhi is called as TH. Okay. Next, here it's called as AH. Here it is called as BH. That means the H antigens, depending on what it is, typhi A or B, paratyphi A or paratyphi B, that's the name as such here. Now look at the thing. Antibodies which are formed against these respective organisms in different patients. Okay, they will not cross react with each other. That means this green antibody is only going to recognize this green antigen, that is TH. That's my example by color coding, just to explain you clearly. Red antibody will not, identify anyone except for this one, that is AH. And same way, blue antibody is this. Is that understood by all of you? So technically, if you find the black antibody and the green antibody, see in the lab testing, it's not going to be green, red like that. I'm just saying in the test tubes, if you see clumps in this and this test tube, you should know that they're talking about Salmonella typhi. Okay, so let me just bring you here. This is what can come in the question. Now pay attention. These are four different test tubes. One is the test tube for the O antigen. Because O antigen is same in all the three, you're not taking three separate test tubes. H antigen being different, you're taking three separate test tubes to figure out who is causing this typhoid. So first, what are you looking for is clumps. If you see clumps, which you are seeing in this example like this, these clumps, you should say antibody against the O antigen is present. That's why the clumps are forming, because they are interacting with each other. Okay, is that clear enough to all of you? This is the thing that should be clear. Now straight away say the person has typhoid. Okay, now we should all be saying the person has typhoid. Next, once you know the person has typhoid, look, right? Look here and there. In the three test tubes, where are you seeing the clumps forming? Now, in this example, the clumps are very clearly shown over here, yes or no? That means, which is this test tube? TH. So now what you should say, first you saw the clumps here, and now you saw the clumps here. So since antibodies are present in both of them, that's why the clumps have formed. So you should say the person has typhoid from Salmonella typhi. TH means Salmonella typhi. On the other hand, if you would have seen the clumps over here, you should have said the person has paratyphi A infection. If you would have seen the clumps here, assume, then you should have called it as paratyphi B. Okay. So that's your Salmonella typhi. Please know, very important, on Wilson Blair media, this guy is going to form black color colonies, jet black colonies on Wilson Blair media. It is a feature of Salmonella typhi. Okay. Next, Vibrio cholera, there is not much I'm going to tell you except for few things. It causes that diarrhea with rice watery stools. Okay. Transport media, again a reminder, Venkat Rama Ramakrishnan and Kerry Blair. How to remember? Short form of Vibrio cholera, V and C. V and C. Okay. This organism shows darting motility. Okay. This is the organism that tends to show what? Darting motility as such. And now look at this important point I'm telling you. This organism shows positivity to this test called as string test. Now at this junction, you don't need the details of this. Okay. Maybe I'll just tell you, listen if you want to. We are just taking a slide and we are adding bile salts into the slide, onto the slide. Then the organism I want to check is added. I'm suspecting it to be Vibrio cholera. Now, what do? Take this thing that I'm highlighting, inoculating loop, take a inoculating loop, touch the slide and slowly retract the inoculating loop. If you see a string forming in this area, it's a confirmation that this is again cholera, because cholera organisms interact with this bile salts in this area, and that is what is going to produce that string. Okay, so that's about it. That's Vibrio cholera. Okay. Next, spirochetes. Please know Treponema pallidum. This is the guy that causes syphilis. Treponema pallidum causes syphilis. Important things to be remembered here. इसमें से जो पूछ सकता है, primary syphilis presents with chancre, which is a painless lesion. And probably you do remember what I'm asking you next. Chancroid. Who causes chancroid? D for Ducrey. Chancre doesn't have a D. Chancroid has a D. D for Ducrey. Haemophilus Ducrey causes that. And the mnemonic was patients do cry. Do cry. We will pronounce it as do cry. Why are the patients crying? Because it is painful. Okay. Secondary syphilis produces condyloma lata. Okay, something similar to the HPV we learned called as condyloma acuminata. Okay, tertiary syphilis mein you should know neurosyphilis. It is going to present as a disorder called as Tabes dorsalis. Dorsal columns would be involved, called as the name of Tabes dorsalis. Okay, so that's about this. Next, hopefully you remember the name of the special pupil here, Argyll Robertson pupil. Argyll Robertson pupil. Okay. Now, how to diagnose this? You should all know this picture for sure properly. This is the dark field microscope. Without adding any stain, it produces a dark background. That is Treponema pallidum. This is the guy that we can stain. Not just Treponema. Any of the spirochetes, we can actually stain on this. Okay, any spirochetes can actually be stained on this. This is the thing, right? Dark field microscopy. Since you see the organism is spiral, they're called as spirochetes. They produce corkscrew motility. Okay, so what is the name here? Corkscrew motility. So be very quick on this. Don't spend too much time. The screening tests available are VDRL and RPR for syphilis. Okay, screening test available are what and what? VDRL and RPR. Only one of them can detect antibodies in the CSF sample also, that is VDRL. Hence, it can be used in diagnosis of neurosyphilis. Okay, neurosyphilis diagnosis, we can definitely use VDRL test. But RPR doesn't have ability to detect antibody from CSF. Hence, we can't use it in the diagnosis of neurosyphilis. Okay, done, doctors? Next, if you talk about this confirmatory tests, they will definitely have the word T or TP in their name. And you should know T or TP simply means Treponema pallidum. Hence, these are the confirmatory tests. These are simply the confirmatory test as such. Okay. Borrelia burgdorferi. This guy causes Lyme's disease. Okay. So Borrelia burgdorferi, it causes the Lyme's disease. Very, very important two keywords. Bull's eye lesion called as erythema migrans, what you're looking at, erythema migrans, and Bell's palsy. Bell's palsy. So bull's eye lesion and Bell's palsy. These are the words to be remembered. Last and the final, you have leptospira. Leptospira comes in your body through rat's urine. Coming in contact directly, indirectly with the rat's urine can give you this infection. And the name of the disease is Weil's disease. Okay, what is the name over here? It's simply called as the Weil's disease. Okay. Now, Weil's disease involves two things: hepatorenal syndrome, as the name says. Because of the liver involvement, it causes jaundice, and another thing is the renal failure. That's something you should all be knowing. So these are the three spirochetes: Treponema, Borrelia, Leptospira. Okay. Now, next is Mycoplasma, a potent organism. Mycoplasma, it is the most common cause of atypical pneumonia. See, pneumonias also clinically are divided as lobar and the interstitial, right? So this one is more or less like interstitial pneumonia, and the most common bacterial cause is this. Now, pure microbiology point, cell wall is absent in mycoplasma. Okay, cell wall is absent in mycoplasma. So then how exactly am I supposed to gram stain it? You can't gram stain this organism because it doesn't have a cell wall. What are you doing in the gram stain? You're staining the cell wall, and that itself is absent here. So this is the organism again that we cannot do the gram stain on. Okay. Next point is this. For the identification of this guy, please remember one last heterophyte test called as cold agglutination test. Cold agglutination test. Now, appearance-wise, this organism shows this thing called as fried egg colonies. Okay, this is described as the fried egg colonies or of Mycoplasma pneumoniae. Okay. Next, chlamydia, I told you it's same as this guy, that is Neisseria gonorrhoeae. I'm just going to cover this shortly once again for you. Look at the name of species, Chlamydia trachomatis. It's telling you I cause a disease from the ophthalm called as trachoma. So this is the guy that is going to cause what? It is causing a disease by the name of trachoma. Okay, so that's your first thing. Next, see here, D2K, they causes genital lesions. The serotypes D2K, they cause genital lesions, which are same as gonorrhea. Everything, including even ophthalmia neonatorum, is same. L1 to L3, they cause this thing called as lymphogranuloma venerum. Okay, lymphogranuloma venerum. This is the thing that you should all be thorough with. Okay. Next, moving on. This is also another agent that cannot be gram stained. Since chlamydia has some viral properties, the best test for this guy is just like a virus called as NAT or PCR. PCR is the best test for chlamydia organism. Okay. Next, coming to rickettsias, keep an eye on this last chart that you have. Bartonella Hensley causes cat scratch disease. Bartonella Hensley also causes bacillary angiomatosis in HIV. And the Q fever, just it can be a one-liner kind of a thing. Q fever, it is by Coxiella burnetii. The Q stands for the query fever. It was like a question, okay? Fever was happening, but the cause was unknown. So it was like a question, who's causing this fever? Hence the name query fever, Q fever, okay? Now, what to be remembered here is all rickettsial infection presents with a rash. The Q fever is the only one that has no rash. That's the thing to remember here. This is the only one not to have any rash. Next, anybody who is a gram negative but not a member of Enterobacteriaceae, it will always be oxidase positive. That organism would be always oxidase positive. That word you should all remember. Okay. Next, virulence factors, if you talk about, see, elongation factor is inhibited again by this guy, just like diphtheria. Diphtheria also does the same. It inhibits elongation factor. Okay. Now, this is the most common cause of corneal ulcers, etc. Some diseases you are seeing here. Now, what is special about Pseudomonas is that it has pigment, properties. Basically, it produces pigments because of which different colors are seen on culture media that I might have made you write. Cyan means bluish color, pyoverdin yellowish green, and pyorubin, ruby is the red one. Okay, so that's about this one here. Next, Legionella ka one or two words. This is the pneumonia that is coming through the AC. And the culture media, if you notice, is jet black because of the presence of the charcoal. Charcoal, BCYE. What is BCYE? Buffered charcoal yeast extract agar. Buffered charcoal yeast extract agar. Okay, so that's about that. Next, this appearance, what you're seeing here, it is basically the one that you're seeing with, this guy called as Bordetella. This is described as mercury drop appearance. Okay, mercury drop or pearl, bisected pearl appearance. This is how we call it. Pearl appearance or this mercury drop appearance is the identification of Bordetella pertussis that causes whooping cough. Whooping cough is by Bordetella. The culture is this one. Okay. Coming to an important organism, Gardnerella. Okay. Next organism is what? Gardnerella. Please know the point. Gardnerella causes bacterial vaginosis. And this disease comes when one of the bacteria called lactobacillus has died. Lactobacillus or Doderling's bacillus, when it has died, we get to see this thing. Okay. You must have learned this Amsel's criteria, and by now you should be thorough in this already. Okay. It's an important question. pH is alkaline in the Amsel's criteria, then only Gardnerella comes. Next, discharge is seen. There is whiff test, which gives you fishy odor when you add 10% KOH, and clue cells. Clue cells. Okay. So those are all the things that you should be familiar with. Campylobacter. What should you know here with Campylobacter? See, one thing, it's a diarrhea that is coming by consumption of chicken and all. So that is something that you should understand. Followed by which, just look at the word, one complication known as Guillain-Barré syndrome. So with Campylobacter, you get to see this Guillain-Barré syndrome, that is a ascending paralysis, meaning weakness from the lower parts of the body to the upper limb. So lower limb gets weak first. Okay, समझ आया सबको? Next, if you notice here, this guy's growth requirements are quite special. Microaerophilic means it requires less oxygen. Capnophilic means it likes carbon dioxide. Thermophilic means it likes higher temperature. Okay. Now, if you do the stain, gram stain, etc., this is what you're going to see, doctors. If you see this bird-like appearance, you should know it is H. pylori or Campylobacter. So Helicobacter and Campylobacter, these are the guys that are going to look like birds' appearance. Okay. Next organism is Haemophilus. Okay. So next one in the list, straightforward. The organism is called as Haemophilus. Now, all you need to know, Haemophilus mein there are two species. Mainly you have to learn Haemophilus influenzae and Haemophilus ducreyi. Now listen to the things I'm telling you. The diseases that are caused by Haemophilus influenzae are same as Streptococcus pneumoniae. Only for the fact that for all these diseases mentioned here, strep pneumo is the most common cause, and this is followed by H influenza. That's the next important cause. Okay, just watch this. Haemophilus, the word itself is telling you it likes blood. So you should know this point over here. You have to grow this guy. That means you should be using blood agar. There are two types of blood agar, as you know. One is chocolate agar and the second is the blood agar. Chocolate agar is the hemolyzed blood. Already the RBCs are broken down in this agar called as chocolate agar. It is the hemolyzed blood. And what happens after hemolysis is two factors get released, factor 5 and factor 10. Okay, factor 5 and factor 10, two things are released here. Okay, if you got this idea. Next, coming to this point here, blood agar, if we talk about, you can't grow this organism on plain blood agar directly. The reason for that is plain blood agar is unhemolyzed blood. Unhemolyzed blood. So now what we are doing to prove that, yes, this guy likes only factor 5 and factor 10 and not the entire blood agar, we are taking blood agar first of all. Okay. It was previously all unhemolyzed blood. It was unhemolyzed blood. So technically, this guy will not grow on this. Okay. This guy will not grow on this. So what we did is, just to prove our point, we put Staph aureus in between like this. And what does the Staph aureus do? Breaks down the RBC. It produces beta hemolysis. It breaks down the RBCs, doctors. And now what happens? Start noticing. Okay. Because in the center, RBCs are broken, but not in the periphery, only in the center part, colonies of H. influenza starts growing like a satellite. Hence, it is known as satellism. Okay, is that understood by all of you? That's what is known as the satellism. So this is the thing that we should know here. Okay. Next, H. ducreyi. H. ducreyi. Okay. It is the one that is going to cause chancroid. D for ducreyi, D for chancroid. Chancre was in syphilis. Chancroid is in, here, ducreyi. The mnemonic is patients do cry. Why the patient's crying? Because it is a painful lesion. Chancroid is a painful lesion. Okay. So that's about it. Please do remember, STDs also, one question will come on this.